സർ നമുക്ക് തുടങ്ങാം. രാധാകൃഷ്ണൻ സർ നമുക്ക് തുടങ്ങാം.
ഓക്കെ.
നമസ്കാരം. SVR കോളേജ് സുവോളജി ഡിപ്പാർട്ട്മെന്റും IQACയും സംയുക്തമായി നടത്തുന്ന മൈക്രോബയോളജി ഫോർ ബിഗിനേഴ്സ് എന്ന വെബിനാർ നമുക്ക് ആരംഭിക്കാം. സ്വാഗത പ്രസംഗത്തിനായി സസ്യശാസ്ത്ര വിഭാഗം മേധാവി ഡോക്ടർ ജയകുമാർ സാറിനെ ക്ഷണിക്കുന്നു.
Thank you, Google sir. Let me discharge my duty. That is to welcome the chief guest and all the participants to the webinar on microbiology for beginners, organized by Department of Zoology in association with IQAC. First, I would welcome the president of this webinar, Dr. Radhakrishnan, respected principal of our college. We are greatly indebted to you for providing us all kind of timely support and creative suggestions in materializing this webinar. I welcome you, ma'am, on behalf of my own self and on behalf of all the participants gathered here. Now, I welcome the chief guest, the resource person of today's program, Dr. E.K. Radhakrishnan, Assistant Professor of School of Bioscience, Joint Director of Inter-University Center for Organic Farming and Sustainable Agriculture, Director of Business Innovation and Incubation Center. Academically, Dr. Radhakrishnan possesses an excellent track record. He was the first rank holder in MSc Microbiology in 2001. He played GATE and CSIR UGC with JRF in 2002. He completed his PhD in biotechnology from RGCD and postdoctoral fellowship in molecular biology from University of Tokyo, Japan. He received many awards in national and international conferences and won Bharat Gaurav Award from India International Friendship Society in 2016. He has research collaborations with many national and international bodies. He has several funded projects and he is a co-investigator in many other projects as well. His area of research include microbiology and nanotechnology. In this field, he has filed for 5 patents and has several books, book chapters, and 123 research publications which include both national and international. He has produced 10 PhDs and 2 of his students have submitted synopsis and currently 7 students are doing PhD undergraduate. We are extremely happy and it is a prestigious moment for Department of Zoology as well as SREE VIDYADHI NSS College for having you as chief guest and a resource person for today's program. I welcome you, sir, on behalf of my own self and on behalf of Department of Zoology, IQAC, and on behalf of all the participants gathered here. I welcome technical support team members, Shri Babin P, Assistant Professor of Mathematics, Srimati Ramya S. Nair, Assistant Professor of Physics. Thank you so much for taking up the charge of technical session. I welcome both of you. Now I welcome my colleague Sri Gogol, who took the initiative for this program, Dr. Peter Pillai, Srimati Shruti C.C., and Dr. Rajesh M.G., who did all the background work in materializing this webinar. I welcome all of you. Now I welcome all the faculties from BIIC department of SREE VIDYADHI NSS College. I welcome faculties.
എന്റെ പ്രിയപ്പെട്ട ഗോകുൽ, ബോട്ടണി ഡിപ്പാർട്ട്മെന്റിലെ മറ്റ് സഹപ്രവർത്തകരെ, വിദ്യാധിരാജ NSS കോളേജിലെ പ്രിയപ്പെട്ട എല്ലാ സഹപ്രവർത്തകരെ, ഏറെ പ്രിയപ്പെട്ട കുഞ്ഞുങ്ങളെ, ഇന്ന് പ്രിൻസിപ്പൽ എന്ന നിലയിലും ഒരു സുവോളജി ഫാക്കൽറ്റി എന്ന നിലയിലും വളരെയധികം സന്തോഷവും അഭിമാനവും തോന്നുന്ന ഒരു നിമിഷമാണിത്. കാരണം നമുക്ക് എല്ലാവർക്കും അറിയാവുന്നത് പോലെ ഈ ലോകത്തിന്റെ തന്നെ ജീവിത ശൈലിയെ തന്നെ മാറ്റിമറിച്ച ഒരു ഒരു വൈറസ് ഉണ്ടാക്കിയ പ്രയാസങ്ങളെ നേരിട്ടുകൊണ്ടിരിക്കുകയാണ് നമ്മൾ എല്ലാവരും. അത്തരത്തിലൊരു സാഹചര്യത്തിൽ മൈക്രോബയോളജിയുമായി ബന്ധപ്പെട്ട ഒരു വെബിനാർ ഇവിടെ സംഘടിപ്പിക്കാൻ സുവോളജി ഒരു ഡിപ്പാർട്ട്മെന്റായി ഇല്ല, അവിടെ ഒരു ഫാക്കൽറ്റി മാത്രമാണുള്ളത്. എങ്കിലും നമുക്കത് കഴിഞ്ഞു എന്നത് വളരെയധികം അഭിമാനം നൽകുന്ന ഒരു കാര്യമാണ്. എനിക്കറിയാം മൈക്രോബയോളജിയുടെ ബേസിക് ആയിട്ടുള്ള കാര്യങ്ങളാണ് സാർ എന്നിവിടെ ഉദ്ദേശിച്ചിട്ടുള്ളത്. എങ്കിൽ പോലും ഇന്നത്തെ പ്രത്യേക സാഹചര്യത്തിൽ ഞാനത് പറഞ്ഞു എന്നുള്ളതേ ഉള്ളൂ. സ്വാഗത പ്രസംഗത്തിൽ ജയകുമാർ സാറ് സൂചിപ്പിച്ചാൽ, ജയകുമാർ സാർ ഇലാബറേറ്റ് ചെയ്തപ്പോൾ തന്നെ നമുക്ക് മനസ്സിലായിട്ടുണ്ട് നമ്മളുടെ ഇന്നത്തെ ചീഫ് സ്പീക്കർ അല്ലെങ്കിൽ കിനോട്ട് അഡ്രസ് നമുക്ക് നൽകുന്ന രാധാകൃഷ്ണൻ സാറിനെ കുറിച്ച് നിങ്ങൾ കേൾക്കുകയുണ്ടായി. അത്തരത്തിലുള്ള വളരെ പ്രഗത്ഭനായ ഒരു റിസോഴ്സ് പേഴ്സണെ നമുക്ക് ലഭിച്ചത് നമ്മുടെ ഭാഗ്യമാണ്. കുട്ടികളോട് എനിക്ക് പറയാനുള്ളത്, പലപ്പോഴും കുറേയധികം വെബിനാറുകൾ നമ്മളിപ്പോൾ നടത്തുകയുണ്ടായി. ചിലപ്പോഴെങ്കിലും കുട്ടികളുടെ ഒരു പ്രാതിനിധ്യം കുറഞ്ഞു പോകുന്നു എന്ന് എനിക്ക് ചിലപ്പോഴൊക്കെ തോന്നിയിട്ടുണ്ട്. ഞാൻ ഓർമ്മിപ്പിക്കാൻ ആഗ്രഹിക്കുന്നത്, വളരെയധികം ഭാഗ്യമാണ് ഇത്തരത്തിലുള്ള അവസരങ്ങൾ നമുക്ക് കിട്ടുന്നത്. എമിനന്റ് ആയിട്ടുള്ള നമുക്ക് ലോകത്തിന്റെ, നമ്മൾ ഇന്റർനാഷണൽ വെബിനാറുകളൊക്കെ നടത്തിയിട്ടുണ്ട്. ലോകത്തിന്റെയും നമ്മുടെ രാജ്യത്തിന്റെയും പല പല കോണുകളിൽ അവരവരുടേതായ തിരക്കുകളിൽ വ്യാപൃതരായിരിക്കുന്ന ഇത്തരം റിസോഴ്സ് പേഴ്സൺസിനെ ഈ ഒരു ഓൺലൈൻ പ്ലാറ്റ്ഫോം ആയതുകൊണ്ടാണ് പലപ്പോഴും പലരെയും നമുക്ക് കിട്ടുന്നത്. ഇല്ലെങ്കിൽ അവരെല്ലാം വളരെയധികം തിരക്കുള്ള ആളുകളാണ്. അപ്പൊ അത്തരത്തിൽ നമുക്ക് ലഭിക്കുന്ന ഈ അവസരങ്ങളെ പരമാവധി പ്രയോജനപ്പെടുത്തണം എന്നാണ് എന്റെ പ്രിയപ്പെട്ട കുട്ടികളോട് എനിക്ക് പറയാനുള്ളത്. ഞാൻ അധികം സമയമെടുക്കുന്നില്ല. ഇന്നത്തെ ഈ വെബിനാറിൽ പങ്കെടുക്കുന്ന എല്ലാവർക്കും ഇത് ഏറ്റവും ഫലപ്രദമായി തീരട്ടെ എന്ന് ആശംസിക്കുന്നു. ഇത്തരം നമ്മളുടെ ഒരു ക്ഷണം സ്വീകരിച്ചതിന് രാധാകൃഷ്ണൻ സാറിനോടുള്ള നന്ദി അറിയിക്കുന്നു. ഒരിക്കൽ കൂടി എല്ലാവർക്കും എല്ലാവിധ ആശംസകളും നേരുന്നു. നമസ്കാരം.
താങ്ക് യു ടീച്ചർ. നമ്മുടെ വിശിഷ്ട അതിഥിയും വിഷയാവതാരകനുമായ ഡോക്ടർ രാധാകൃഷ്ണൻ സാർ BIIC ഡയറക്ടറും MG യൂണിവേഴ്സിറ്റി മൈക്രോബയോളജി അസിസ്റ്റന്റ് പ്രൊഫസറുമായി ഇദ്ദേഹത്തിന്റെ അക്കാഡമിക് അച്ചീവ്മെന്റ് ഒട്ടനവധിയാണ്. മൈക്രോബയോളജിയിലും നാനോ ടെക്നോളജിയിലും അതിവിദഗ്ധനായ രാധാകൃഷ്ണൻ സാറിനെ മൈക്രോബയോളജി ഫോർ ബിഗിനേഴ്സ് എന്ന വിഷയത്തിൽ വിഷയാവതരണത്തിനായി ക്ഷണിക്കുന്നു. രാധാകൃഷ്ണൻ സാർ
തുടങ്ങാം. തുടങ്ങാമോ?
തുടങ്ങാം.
Okay. So first of all, I express sincere thanks to the organizers for inviting me for this particular webinar, and especially I express sincere thanks to the principal and also the coordinator. And I think mainly the participants are like BSc students. I think so, isn't it?
Sir, BSc MSc കാരുണ്ട്.
Okay, BSc in botany and zoology maybe.
Okay, ബോട്ടണിയും സുവോളജിയും ഉണ്ട്. പല കോളേജുകളിൽ നിന്ന് ജോയിൻ ചെയ്തിട്ടുണ്ട്.
Okay. So we will just go through the slides. കാണാമോ? സ്ലൈഡ്സ് കാണാമോ?
കാണാം സർ.
Okay. So whatever the subject, whether it is microbiology, botany, zoology, or biochemistry, or biophysics, or anything like anatomy, physiology, whatever it is, ultimately all these subjects deal with the life or living organism or living system. And when we think or when we study or when we consider anything about life, we should start actually with the evolution of life. So evolution of life, as we all know, it's considered to be originated. Life is considered to be originated somewhat 3.5 billion years ago. And if we go back to the time and origin of life, what we can see that the first established form of life was in the form of microorganisms, and later, over the period, other organisms evolved, and the life or living organisms as we today in the form of plants or animals or whatever it is the final stage of that evolution that we can see today. And one important thing with respect to this journey of life or the evolution of life, is that the first established form I mentioned earlier, that it was in the form of microorganisms, and later we could uh, identify the evolution in the form of diversity or difference is there, because as we know, plants are entirely different from animals, and animals again, there are very different types of species are there. And but still, during this evolution of life and establishment of different living forms, the microorganisms are playing some important role in the existence, functioning, and to make the other living systems in a perfect manner. Because why it is that much important, that we will just go through some of the basic things. And especially, so when we see microorganisms, the first question comes, where it is present. That means, what about its whether it is something different from us, or whether when we study something, so we should have an idea about where it is present. So basically, it is present everywhere. So they are extremely abundant and diverse in nature. And the image shown here is the different magnifications of magnification levels of microorganisms which are present on the surface of a pin. So just a simple object or simple material mentioned here is that even in the tip of a pin, there are thousands of organisms are present. So that this is just to give you an idea that these microorganisms are present everywhere, especially for those who just start to study these things, that this idea is very important. So they are present everywhere. They are present everywhere. So why they are important, that we know. And so far, based on the understanding of these microorganisms, why they are important means they are used for different purposes, especially for industrial applications, and they are clinically or medically important, that we know, especially now during this period. We are all we are all familiar with, we are all we know well than anything else earlier, because even though we study in textbooks some disease or pathogen or disease transmission and its impact on human, and in most of the cases we may think that these pathogens are something maybe for someone only, so it may not be applicable to us like that. But now this situation is making us to be aware that it can get enter, or it can cause disease in any person. So medical importance is there, and agricultural importance is there, because there are so much of applications in the agriculture, environmental application, food, and many sectors are there. And even so, when we consider the developments in microbiology, it also when we consider the different developments or at the same time applications, so even synthetic bacterial cells were generated. That means you can see the reports here, reports from Ecuador Institute, and they already reported the synthetic bacteria. Synthetic bacteria in the sense they just created the genome created artificially, and this artificial or synthetic genome was introduced into MG cell, and it later became a replicating cell as just like a normal cell. So that much of understanding of microorganisms. So when we see the basic aspects, how these microbiology evolved, you can see the different steps or different processes or different findings which made the foundation of the science. Because unlike other branches of science, for example, like plants or animals that we study, the presence itself is not a problem here, because these organisms are microscopic in nature. That means we cannot see them or we cannot observe them directly with our naked eye. So the first basic requirement was development of lens system. So So this lens system developed by Anthony van Leeuwenhoek, later contributions from Louis Pasteur, Robert Koch, and later these developments were merged with the developments in molecular biology, and now we are moving towards this stage. But now the question is, so earlier there was a time that means it was unknown. That means the presence of microorganisms itself was unknown because of the lack of instruments or methods to see the organism. And later, with the development of lens systems and microscope and other advanced study tools, it became known that these organisms are there. We can study them. But now the question is, up to what level that we know, that is a real question. So, in the case of the corona itself, that we know that the viruses, already it's known that the viruses are present. But when that particular pandemic situation came, now we know that what we know is only very limited thing, because the entire world is struggling to manage these organisms. So even though we know something about virus, we know something about its structures, we know something about that two very simple and very basic thing that we know. And with that knowledge itself, we are not in a stage to manage even a single virus, because we already the studies or findings or research and developments focus on many pathogenic organisms, and we could identify methods to manage many of them also, and due to through the introduction of antibiotics, through the vaccination, through the other control measures and like that. But in the case of these newly emerging viruses, again, it became that we have to start from zero itself. That's the situation. So even though what we know something about the microorganism, when a new challenge comes, it becomes totally just we have to learn many things from them. So that's a real challenge. And this Anthony van Leeuwenhoek, that person, so he made the important contribution in developing the lens system, because he is the first person to observe living microorganisms, living microbes. So because he developed a magnified lens, he developed a lens system which could magnify up to 300 times. And later, the contributions of Louis Pasteur, and so he identified or developed some very important steps or principles, and developed his findings formed the very basic steps towards the development of this area. He showed microbes are the one which is responsible for the fermentation and spoilage. At that time, it was considered that it can be due to somebody told that it is due to some living system, others mentioned that it is not, something like that. So there were much controversy were there, but actually the scientific basis for the same was provided by the Louis Pasteur, and like that he developed many contributions are there. Later, another important contribution is by Robert Koch, and he actually focused on pure culture method, and he could identify the organisms which cause many disease also. And he developed a so-called Koch postulate, a series of experiments to predict whether a particular organism can cause a disease or not. So at that time, it was it is very much important. It was very much important to identify or develop procedure or develop method or steps to identify whether a particular organism is a pathogen or not. So he developed the Koch postulate in such a way that if you have to consider a particular organism as a pathogenic one, so it should be isolated from a pathogenic. It should be isolated from a diseased animal, that is shown here. So if this organism is to be considered as a pathogen, it should be isolated from a diseased animal. It should be isolated in pure culture. And when this pure culture, the organism in pure culture, if it is introduced into a healthy animal, that due to the presence of this organism, the healthy animal should change into a diseased condition. And from this diseased animal, again, we have to isolate the organism, and this organism, if the isolated organism from the second animal is the same as that of the this organism, if both are same, means we can confirm that this particular organism is a pathogen. So this is very important step, and still it is an important step to identify that or confirm an organism whether it is a pathogen or not. So based on that one, now we will just go through the relative size aspects of microorganisms. So as we all know, their size is in micrometer range. They are microscopic in nature. So this is just a comparison of sizes. This is a eukaryotic cells, and you can see it's a size-wise comparison. With some of the selected bacteria, Staphylococcus, E. coli, and viruses are also given. So you will get an idea about the size comparison. Basically, the microorganisms are in micrometer range. The size is in micrometer. So how this micrometer is compared with other units here, you can see a scale here. So here you can see that this is most of the microorganisms, especially bacteria, comes under this range where it is a range of a light microscope, and this is a range of human eye. That some examples are given that you can see. This is a range of light microscope, and here most of the bacteria will come at this range, and this is a range of electron microscope where the viruses and the other components will come. So you will get here. This is just to give you an idea about the size-wise features or comparison of microorganism. So when we say microbiology, again, it includes many components are there. As we know, viruses are there, then viroids are there, prions are there. So then bacteria, archaebacteria, and also based on eukaryotic organisms are also there, like protozoans are there, and other organisms are there, because all these considered under the microbiology. So viruses, as we know, they are importance that we know. Viroids are their RNA molecules, which are mainly found to cause some infection or disease in like plants, and prions, they are proteinaceous infectious material which cause some disease in humans also. So this is just like a different components or different members of or what are the organisms there. So coming to the structure of a particular prokaryotic cell, now we not just they have a surface appendages. Appendages means there are surface structures are present. These are meant for different purpose like movement, for motility and interaction with other organisms and so on. Then cell envelope is there, which is basically meant to protect the cells and for mediating some biochemical functions, and the cytoplasm, which is having all the other components. So what are the basic steps involved in microbiology? This involve what we can say as 5 I's. I means inoculation, incubation, isolation, inspection, and identification. So this is just for you to know something about the basic steps involved. Inoculation is introduction of the collecting the microorganism from a source, or introducing this microorganism from one source to a experimental media. That process is inoculation. For example, if we want to study about the microorganism in the water, water will be the inoculum. Water samples collected will be the inoculum. If we want to study something from a human sample, maybe blood may be the sample, or like a sputum may be the sample, or anything, any material that we use to study the presence of microorganisms in that particular sample from the inoculum, and it is added in a particular media and it will be incubated. That means it will be kept under controlled condition based on our objective. That means sometimes it will be kept under 37 degrees Celsius, sometimes it may be at 28 degrees Celsius. That will depend on our objective. Then isolation, inspection, and identification. So after the culturing, that this is a typical example that you can see that this is a particular media where it is exposed to, or it is inoculated with some samples. Maybe I think it is from some soil sample something. So you can see that different so-called colonies are there, and these colonies are marked in different ways based on their many features, like their size, their shape, their pigmentation, their transmittance or light transmittance, and many properties are border, and many features are. So each of these different colonies represent different organisms. So here in this particular figure, you can see both bacteria and fungi also. So this is just like the process that I mentioned is again summarized here. So this is a particular inoculum where different microorganisms are there. It is introduced into a media after the incubation. So we can see that from one organism it will multiply and form a particular colony. So based on the number of colonies formed or the types of the colonies formed, we can identify the number of organisms present in that original material, And here, as you can see, only two different organisms, two different types of organisms are there. So that only two different types of colonies we can observe here. So this is the method for when we when we isolate particular colony also, it is likely that sometimes it may be formed from one organism or from another organism also. So there can be so that it should be purified to make it sure that this particular colony is formed from a single organism. That is called culture purification. And here that process is there. You can see that this is petri dishes which are filled with media, and this media, this process of isolating this or making the culture in a pure stage is called a streaking. So you can see that this is a bacteriological loop. It can be separated in diverse ways, so that ultimately we will result in the isolated colony. So in this case, these colonies can be considered as a pure form. Hello, sorry. Hello, sorry. അതെ അതെ റേഡിയേഷൻ ആണോ? ഞാനൊരു ക്ലാസ്സിലാണ്, അപ്പൊ ഒരു 3:30 ആകുമ്പോൾ ഞാൻ ചെന്നോളാം. ശരി ശരി ശരി. So I think it's this is clear to you that this is a process of isolation is called as streaking. And here the meaning of this process is that when we take a particular inoculum here, it will have so many different cells are there. And during this process, it is considered that the organisms will be isolated. So this isolation will be made in a correct manner in such a way that the different cells will be separated. And ultimately, for example, this colony that we can see after the incubation, actually it is formed from a single organism that might have isolated during the process of streaking. So this one is considered as a pure culture, because when we analyze all these regions, we can see only one type of colony, because so that it is clear that it is a pure culture. And in this case, this particular colony is formed from a single organism. So when we incubate in the media over the period of incubation, typically it is for in the case of 37 degrees Celsius. For generally we will use for overnight incubation. And overnight incubation, it will grow and multiply, and a large number of cells will be formed, and it will ultimately result in the visible form of the colony. So these microorganisms, just we are discussing more about bacteria. So they exhibit diversity in different aspects, and how this diversity is there, and how we can study this thing. So just now we studied about isolating the organism. So now to identify their morphology, the process is called simple staining is there. So the simple process of simple staining is used just to give the morphological feature of the organism. So this is an experimental procedure that you may be familiar with this one. So this a series of steps are there. Ultimately, what we can see is the morphology of the organism. So based on the morphology, you can see these are the normal morphological features of bacteria. It can be in a different size and shape. Both are there. It can be generally in the coquet-shaped or round-shaped or rod-shaped, or in the case of a spiral shape. The coquet-shaped structure itself, it can be in different forms. Different types of arrangement is there. Like it can be diplocoque, or it can be tetrad, or it can be sarcine, it can be streptocoque, or it can be staphylocoque. But each of these arrangements will be unique for a unique group of organisms. So that gives an idea about its diversity. And at the same time, regarding the rods itself, the rods, you can see so many diverse forms of rods are there. All these are rods, and these rods again can associate in like chains, or there can be cocobacillary, and this different shapes are there. And in the case of the presence of granules, and the spores will be varying. Then their branching rods will be there. Then again the spiral shaped one, it can be again in different forms, like Vibrio, like a comma-shaped organism, or spirula. And in spirochetes, you can see the number of spirals or coils in these are different. Then there can be many different. So this is just to give you an idea about the diversity with respect to the morphology of bacteria. bacteria. And another important thing is, so bacteria, they are categorized mainly into gram positive and negative one based on their response to a particular procedure called gram staining. And this is one that you can see here. This procedure, it's again a series of steps where it is treated with different dyes, and ultimately that you can see that it is categorized into two different forms, either gram positive or gram negative one. So this gram positive and the gram negative one, they are the basic, again, a process for the identification. So based on the color they are retained after the staining reaction, and so it will be gram positive or negative. And just like that, different staining methods are there. This is giving an overall idea about different staining methods, like the simple stains used to identify the morphology, differential stain like gram stain to distinguish different organisms, whether it is gram positive or not. Then special stains for studying capsule, then studying the for inclusions bodies, and then for microbacteria, and like that, variety of staining methods are used to identify different properties of microorganisms. So based on these gram staining, they are categorized into whether it is gram positive or gram negative, and there are organisms without cell wall also, then bacteria with a unique, chemically unique cell walls are also there. So how the gram positive and gram negative, how it will look, how it will look like. So this is a simplified representation that you can see that in the case of gram positive organism, the so-called peptidoglycan is present and it is thick in nature, but in the case of gram negative one, that you can see that there is a thin layer of peptidoglycan is there, and external to the or outer to the peptidoglycan there is a so-called lipopolysaccharide layer is there. So this is a basic difference in these two organisms. That's why, or that's the basis of its difference in differential response to staining procedures. So this again a cartoon representation that you can see that this is a set of gram positive one and this is gram negative one. And in the case of gram positive, there is a thick peptidoglycan layer is present, and here a thin peptidoglycan, but here outer lipopolysaccharide layer is present. So this is a basic difference. And this peptidoglycan again that we mentioned, peptidoglycan is present in the cell wall of bacteria, and it is composed of N-acetyl glucosamine and acetylmuramic acid, and they are connected just like as you can see that one after another they are connected just like a chain. And this is just like a beads in a string. You can see that this is N-acetyl glucosamine, this is N-acetylmuramic acid, just like that it is there. And these different chains are interconnected also. That means provide more structural rigidity to this one. And these two are connected by beta 1 for glycosidic linkages also. So this is a basic aspect about the peptidoglycan linkage, and this is about the already we mentioned about the structures for which are essential for its mobility or motility, and this is about the flagella that we discuss about flagella here. So you can, and how the flagella looks and how it grows, that is shown here. That you can see that this is a growing endoflagella is shown in this figure. That you can see that it is formed of subunits called flagellin, and these flagellins are transported across the growing tube and it is added to the growing ends. That's why you can see that it is coming out of that growing end and it is getting polymerized, or it is added to the growing end, just like that also. And this is the basic features of flagella. It is getting integrated into the cell wall, and there are many complex structural features are there. And this is the outer one that we can see as a filament. There is a hook region and a basal body is also there. And again, the flagella is a general structural like feature that we shown here. So you can see a single flagellum here, but this flagella, the number of flagella present in the organism can vary with respect to the particular species of organism. That can be a feature. And this is a typical representation of types of flagella which are present in bacteria. And this can be a single flagellum, group of flagella at one end, flagella at both ends, or a flagella which are present throughout the surface of the organism. So this is just to give you an idea about this flagella. It can have different types of distribution. That is a unique feature of each of these organisms. So this is with respect to, so what we mentioned here on is with respect to diversity. So when the morphology is there is diversity, then flagella is there is diversity, and the metabolic aspects also. So as we know, all organisms require a source of carbon and energy for their growth multiplication. So there is a major nutritional requirements are there for all the organisms. So bacteria are very diverse in the sense that they can use variety of energy source, variety of carbon source, and there can be autotrophic or heterotrophic organisms. So variety of carbon and energy source can be used by this organism. That again indicates its ability to adapt to in any environment. And all these things we have to correlate to their origin as a first established living form, because they are adapted. That's very important. They are adapted to live in any environment than any other organism or any other is, because they were the first originated form, so they have the all the properties to adapt optimally to the environment. So this with respect to carbon and energy source. With respect to again for oxygen requirement, you can see there are different types of organisms are there. Some can grow in presence of oxygen, some in the absence of oxygen, or some in case of presence or absence, or like that. There are organisms are there, and another metabolic diversity here is the microbes which are present at extreme conditions. That you can see that bacteria. There are hydrogen oxidation bacteria, carbon monoxide oxidation bacteria, ammonium oxidation bacteria, nitrate oxidation bacteria, so like that, and oxidation bacteria, methanogenic bacteria. So all these indicate their metabolic diversity, and they are performing very important functions in different environment. And they are also well known for their presence in the extreme condition that we cannot imagine. So there are so-called extremophilic organisms are present. So this is some of the natural environment where they are present. So extreme cases of temperature, and when they are those organisms which live at this extreme range of temperature, they are thermophiles which grow at a high temperature. Then psychrophiles are there at the opposite end. That means they are the organisms which can live at very low temperature. So these are again adaptation to live in different extreme temperature. So there you can see that thermophiles in the isolated from the natural environment, and the psychrophiles from other environment. Then there are chemical extremes are also there. Bacteria there which can be acidophilic organisms or alkalophilic organisms or halophilic organisms. So they are adapted to live in any environment. That is a basic thing. So this is, you can see acidophiles with a very low pH range, and halophiles are there with a high salt condition, and their survival in. And another important group is even bacteria there which can tolerate or tolerance to radiation. So extreme ionizing radiation resistance is there. For example, Deinococcus is an important one, which is well known for its resistance to radiation also. And these are some of the natural adaptation. But at the same time, there are these are another point of view where that means due to the introduction of what we can say that due to the introduction of antibiotics. So there is we are using these antibiotics to kill the microorganism, but at the same time, microorganism is also developing methods to over. Organism which cause disease to us. But at the same time, from the organism point of view, they should have the method to resist this antibiotic. Then only they can exist. So these parallel challenges, evolution, many things are progressing. So this is another important aspect of this one. So these microorganisms are involved in many important features or many natural processes even that we are not familiar with. So there are studies that there are processes like rain-making bacteria, the so-called, they are related in some way to the rain also. So there are bacteria which are, so you can see that geobacter, which is found to have important application as electric microbe. Then like that. So now to the another challenge here is that when we conduct a microbiological study, the question is, so in the first in one of the first slides, again, I mentioned that there was a period that it was unknown. Then later it became known. Another question now is up to what level really we know. That's a real question in another many contexts. That question is there. So this is one important thing in the case of microbiology, that this one experiment conducted earlier. So this from this experiment that this observed result is called a great plate count anomaly. So in this case, you can see that here this is a seawater sample, and this is a bacteria like which are growing in a media collected from the seawater. So when they compare the total number of bacteria from this one and compare the total number of organisms present in the original sample, the number was not matching. So not matching in the sense there was huge differences there, that you can see that here only hundreds of organisms could be identified from ml, but you can see the number here. So from among these organisms, only this many organisms could be isolated in the plate. So the question came how or why it is not possible to culture majority of the organisms. So here comes another important thing, that vast majority of the microbial world is still unknown because they are not culturable. And it is considered that more than 90 to 99% of organisms are not yet cultured. What we know is only from limited number of organisms. What we studied is only from limited number of organisms, but still many complex and important things are there. And so how to study the unculturable organism? There came another area for studying unculturable organism, that is called metagenomics. And this metagenomics is a branch which deals with not studying the organism by culturing it, but studying the total organism. For example, if we want to study the microorganisms in a particular soil sample, in the case of metagenomic approach, what we will do is that we will isolate the total DNA from the soil. In the sense, the different organisms present in the soil, the DNA present in different organisms which are actually present in the soil will be isolated without culturing. So this, the crude soil sample will be collected, it will be processed, and the total DNA will be isolated from that sample, and it will be used for the study. So this is a metagenomics, where the application of modern genomic technique to study microbial community. So now the initial slides I discussed mainly about the contributions about pure culture, and that period the studies on pure culture was very important at that stage, because at that stage majority of these challenges were with respect to pathogenic organisms, especially those cause different types of diseases. So in that case, but to identify the organism, to identify its features, to study its clinical importance, and to identify methods to manage those organisms, it is important to have the pure culture. So our studies initially focused mainly on pure culture or single type of organism. But now the trend is moving towards studying the microorganisms in a community. That means group of microorganisms, because that is getting more importance now. Because what we consider is that the microorganisms, when they present in a environment, that is more, there is much more things that we have to learn from them. So from this one, the term is coming, like the microbiome, the term microorganism to microbiome. So we are more studying the microbial community. So the community can be anything like a natural community or a host associated community or artificial community. So this is a metagenomic study, typical step involved, that you can see this particular soil sample and the isolation of DNA from the soil sample and processing it, and isolating the total DNA and processing it and sequencing the DNA and studying their features. So from this one, two methods are used. It can be a sequence-based metagenomics or function-based metagenomics. Sequence-based metagenomics is used to give some idea about its identity, to identify which organism is there. Function-based means based on the sequence that we can get, it can be translated into protein, and from the protein sequence we can have prediction about its function. So from this one, many important things are coming, like some of the contributions that you can see that in this case, biosynthetic pathway can be reconstituted from metagenome. Because in this particular study, what they did was, you may be knowing, the marine source is one of the important sources for bioactive natural products. So in some of the study that they could find that in that marine source they studied actual bioactive compound. A compound is produced by the microorganism, but this organism was not in a culturable stage. It is not. It is an unculturable organism. But they isolated the total DNA and they could constitute the biosynthetic pathway. That means different genes involved in this biosynthetic pathway were isolated from the metagenome and they were like introduced into different vector system and ultimately into another model organism and then produce that particular metabolite. So these are some of the applications of these unculturable organisms. Then novel enzymes can be identified from this one. So from this one we are coming to the microbiome. So microbiome means the total microorganisms which are present in a particular site or a region. So if we analyze the microorganisms which are present in human, it is called a human microbiome. If we study the microorganism present on the skin of human, it is skin microbiome. If it is present in the like a mouth or oral cavity, it is oral microbiome. If it is in the intestine, it is intestinal microbiome. If it is plant, it is plant microbiome. If it is like earth, it is earth microbiome. So now the focus is more towards studying microorganisms which are present at a particular region. So in the case of human microbiome, you can see this type of international research progress also in the case of human microbiome project. So you might have heard about the human genome project earlier that completed some years back, and now there are like a human microbiome project is also there regarding which is related to identifying the microorganisms present in different parts of the body. So that is very important. So initially in the first stage, we focused mainly on the pathogenic organism, but human system is having large number of organisms which are highly beneficial. So just like I mentioned in some of the introductory slides about the surface of a pin, you can see large number of organisms, but it is not a living material. But if these organisms are present in a living system, living material, living surface, it can have much more meaning because these organisms may be related to some of the functions or of the particular organism, whether it can be human or plant, because again we have to go back to the evolution of life, the first established possible. So for humans to survive, for humans to live, to grow, to be healthy, for all these things, these microorganisms are very important. Beneficial organisms are important. So it is considered that all of us have a large number of microorganisms, and some of the microorganisms can be common to all of us. For me, for you, and for others, there are some organisms which are common. So they are core human microbiome, but most of the organisms can be variable also because we all have a difference in many things. The food that we eat, our physiological nature, our the time, temperature, or food nature of food that we eat, the age, and many things are there which can have a impact on the type of microorganisms present in a particular organism. So there are some studies are going on where the group is saying that the microorganisms present in the human can have a determining effect on many features of human. Like here it is saying that getting personal with the bacteria, that means even a behavior of a person can have some basis to microorganisms present in humans. So like that, these microbes are present on different parts of, but this much of contribution is by the microorganisms associated with the human. So that is the case with the human or plant or any other system. So this is considered that the microorganisms associated with the human, it can be a second genome of human, or it can be a second genome of plant in the case of plant. So this is an extended genome. Many things are coming. So at the time of birth itself, the microbial association starts. So all the stages. So this is just to even the first moment of birth itself can have a determining effect on which type of organism should be associated with the newborn, the nature of delivery, or the type of food given to the newborn, and so many initial stages and steps itself can have a determining effect on the type of microbial association. So this is some of the studies indicating different microorganisms present in the human, and there are some interesting things are also coming like that. Even a very small change in our physiology, our biochemical process, or our emotional status can even have a determining effect on the microorganism also, because there are fine association, very sensitive association is there with respect to human and microorganism. So even a change in slight change in temperature or slight change in our physiological process can change the microbial association. So another thing is that the gut flora and the brain, the functioning of the brain is also considered to be have some relation with the microorganisms and functioning in the intestine. So this is you can see that here there is a gut-brain axis is considered to be there, and it is considered to be kept under normal by the functioning of the healthy microflora present in the intestine. So when it is changed, this healthy interaction is changed or healthy balance is changed, we will be in a stage called a stressed condition or diseased condition. So the this healthy functioning is supported by the beneficial organism. So obesity always, obesity, another important issue, is considered also to be related very much to the microorganisms present in the intestine, and it is also considered to have the possibility that wherever we touch, so there are unique organisms can be present in each of us, and wherever we touch, we may be leaving some of the organisms present on our surface on that particular material also. So it is can have some future application in forensic application also. So these beneficial organisms are used for different health benefit also. You may be knowing that the application of probiotics, then different types of products are there. So these are examples that you can you can get an idea that these beneficial organisms can be used in many ways. And you can see here prebiotic. These are some of the materials which are given to enrich the beneficial organism. So these are also there. So there's a slide about metagenomics of human colon microbiome. This study indicates that our digestive process, because we are consuming a variety of food, and our digestion process is supported very much by the microbial enzymes, not by our enzymes, because we do not have all the enzymes to digest the complex food material, and some of these enzymes are provided by the associated microorganisms. So this is just gave you some idea about humans, but the same thing is applicable to plants also. So this just give you an idea. So we are all familiar with the diversity of plant material, and we know that all these different many things as we generally consider like size, shape, color, or whatever it is. But it is now emerging that the type of microorganisms present in this can also vary, because they are performing very important function in the growth and existence of plants. So the plant microbiome, because as we know, the plants, they live in the soil, but we cannot say that it is in the soil, but in another way we can say that it is there in a pool of microorganisms. That will give much more meaning. So because even in the case of leaf, you can see that these are some of the microorganisms. They are performing very important functions for the function, supporting the function of leaf. So in the case of root, again, large number of variety of microorganisms are there which are performing important role for the mobilization of nutrient from soil to the root, and thereby making the plant. So these are there. So this indicate that there are entry of some microorganisms into the plant. That's there. So plant microorganisms are present on the surface of the plant and also within the plant. So plant is permitting some of the organisms to enter into the plant. So the plant is permitting some of the soil microorganisms to enter into the plant. So why it is permitting? Because these organisms are essential for the growth and functioning of plant also. So this is another important application. So this microorganism, because we can see that this microorganism, this is particular organism, this is a plant cell. So the functioning of the plant is very much influenced by the microorganism, because they produce different types of plant hormones are produced by microorganisms. For example, here you can see IA producing microorganism. This is a large number of organisms are there which produce plant hormone and support plant growth, and they can regulate the hormone in the plant also. So ethylene content is regulated by the enzymes produced by microorganism. They perform important role in mobilizing the nutrients, and also very important role in making the plant system resistant to pathogens, so inducing the resistance mechanism. So these are very important things where many important applications like biocontrol application, resistance induction, and growth promotion, and many applications. So when we all these organisms are performing many diverse processes which are very essential for the survival of the plant. And plant minus this organism means the plant cannot exist. That because the importance is that much. So we know that the so-called many processes are there, like phytoremediation, like many terms are there, like phytovoltaization, phytoevaporation, phytosimulation. All these processes, processes are some way or another way supported or by the functioning of microorganisms. So these microorganisms are present within the plant also. This is another important thing that gives you an idea, because medicinal plants that we all know, because these are some of the well-known medicinal plants. These are some of the well-known medicinal plants which are producing very important metabolites. These are anti-cancer metabolites: taxol, camptothecin, podophyllatoxin, etc. So these are very important anti-cancer compounds, and these are the plants which produce this anti-cancer compound. But recent studies indicate that microorganisms, especially fungi, which are present within the plant, can also produce this one. For example, in the case of Taxus brevifolia, one organism is isolated called Taxomyces andriani, and this organism isolated from Taxus, this is a plant. From this plant, or this fungi, this fungus is present within the plant, or this fungus is isolated from the plant, and this fungus which was isolated from this plant is able to produce Taxol. So like that, in other plant studies also, it is found that these very important phytochemicals or bioactive components which are isolated from all these plants are also produced by the fungi which are isolated from the same plant. So the question comes whether the plant is actually producing this one, or fungi is actually producing this one, or both are producing this one. So anyway, the things are becoming more important in the sense that the existence or functioning of humans or plants or any system is basically determined by the microorganism. So this is another example that you can see that as taxol is one important bioactive metabolite. So large number of fungi were isolated from different plants, and you can see that in many plants these isolated fungi were found to produce taxol. So this is a very important thing. So here we are concluding in the sense. So what I tried is just to give you an idea about the importance of microorganisms and why it is important and how it is important. And from starting from the pure culture and isolating these organisms from through very simple stage. And what we learn through microbiology is basically we are studying about ourselves or about the life itself, because existence of human or plant or any other system is ultimately determined by the microorganism. So any process, anything can be related to this microbial role is there. So actually we have to learn much things from microorganisms itself to identify many solutions to manage the challenges like health of human or health of plant or agricultural productivity or disease resistance of human or whatever it is, can be managed through a detailed identification, detailed analysis about the functional involvement of microorganisms with other organisms. Thank you all. So if there is any questions, you can ask.
താങ്ക് യു സർ. എന്തെങ്കിലും സംശയമുണ്ട് സാറിനോട് ഇപ്പം ചോദിക്കാവുന്നതാണ്.
രാധാകൃഷ്ണൻ സാർ.
യെസ്.
സാർ ഒരു ഡൗട്ട് ഉള്ളത്.
പ്ലീസ്.
ബാക്ടീരിയ സ്പീഷീസ് കൺഫർമേഷൻ, ബാക്ടീരിയ പുതിയൊരു സ്പീഷീസ് ആണ് എന്ന് കൺഫേം ചെയ്യാൻ 16 SRRNA സീക്വൻസിങ് അത് ഇനഫ് ആണോ?
അതിന്റെ ഒരു ഫസ്റ്റ് ഒരു പ്രിലിമിനറി സ്റ്റേജിൽ നമുക്ക് അത് വെച്ചിട്ട് പറയാം. ബട്ട് ബേസിക്കലി നമുക്കിപ്പോൾ അതിന്റെ ഏറ്റവും ഒത്തന്റിക് ആയിട്ടുള്ള ഒരു കൺഫർമേഷൻ നമുക്ക് ഹോൾ ജീനോം സീക്വൻസിങ് ചെയ്യാവുന്നതാണ്. ദാറ്റ് ഈസ് നൗ ബിക്കമിങ് മോർ കോസ്റ്റ് എഫക്റ്റീവ് ഓൾസോ. So, but from the 16 sequencing, we can have a preliminary information. It is likely that it can be a novel species or a new species.
ഓക്കെ സാർ. രണ്ടാമത്തെ ഒരു സംശയം, നമുക്ക് പലപ്പോഴും റിസർച്ച് ചെയ്യുമ്പോഴത്തേക്ക് പുതിയ സ്ട്രെയിനുകൾ ബൈ സീക്വൻസ് അനാലിസിസ് ചെയ്യുമ്പോൾ പലപ്പോഴും കാണാറുണ്ട് ഒരു പുതിയൊരു സ്ട്രെയിൻ ആണെന്ന് കാണാറുണ്ട്. അപ്പൊ ഇങ്ങനെയുള്ള സ്ട്രെയിനുകൾ നമുക്കൊരു ലോങ് ടേം ഡ്യൂറേഷൻ ബേസിസിൽ എങ്ങനെ സ്റ്റോർ ചെയ്യാം?
അത് കൂടുതൽ നമ്മൾ കൾച്ചർ മെയിൻ്റെനൻസ്, കൾച്ചർ സ്റ്റോറേജ് എന്ന് പറയുന്ന കൂടുതൽ നമ്മൾ എന്താണ് different types of preservation or storage methods can be used, and basically in the laboratory the basic method is based on low temperature store ചെയ്യുക, especially in -80 ലയോഫിലൈസ് ചെയ്തിട്ട് സ്റ്റോർ ചെയ്യുക എന്നുള്ളതാണ് one of the important one.
ഏതെങ്കിലും ഏജൻസിയോ റിസർച്ച് സെന്റർ ഉണ്ടോ സാർ? നമുക്കിപ്പോ കോളേജുകളിൽ പുതിയ സ്ട്രെയിൻ കിട്ടിക്കഴിഞ്ഞാൽ നമുക്കത് ഡൊണേറ്റ് ചെയ്യാൻ അല്ലെങ്കിൽ ഡിപ്പോസിറ്റ് ചെയ്യാൻ പറ്റുന്ന ഏതെങ്കിലും സ്ഥാപനങ്ങളുണ്ടോ?
So based on the importance of the strain, for example, if that strain is entirely novel strain, or if that strain is like a high yield, and any unique property there are national centers are there where you can submit. അപ്പോൾ നമുക്ക് ഇംടെക്കിൽ സബ്മിറ്റ് ചെയ്യാം, അല്ലെങ്കിൽ അതുപോലുള്ള. പക്ഷെ അത് എല്ലാ ഓർഗാനിസവും അങ്ങനെ പറ്റില്ല. there should be some extra feature or extra ordinary ആയിട്ടുള്ള ഓർഗാനിസം ആയിരിക്കണം. There are many culture collection centers are there.
Okay, thank you, sir.
ആർക്കെങ്കിലും സംശയം ഉണ്ടെങ്കിൽ മൈക്ക് അൺമ്യൂട്ട് ചെയ്തിട്ട് സംസാരിക്കാം, സാറിനോട് ചോദിക്കാവുന്നതാണ്.
ഒരു ഡൗട്ട് കൂടി സാർ. നമ്മളിപ്പോ ഒരു ഉദാഹരണത്തിന് ഒരു ഫോസ്ഫേറ്റ് സോലിബിലൈസിങ് ബാക്ടീരിയ സ്ട്രെയിന് കിട്ടിയെന്നിരിക്കട്ടെ. അപ്പൊ അതിന്റെ ആക്ടിവിറ്റിയെ എൻഹാൻസ് ചെയ്യാനായിട്ട് നമുക്ക് എന്തൊക്കെ മ്യൂട്ടേഷൻ സ്റ്റഡി പോലെ മറ്റെന്തൊക്കെ സ്ട്രാറ്റജീസ് ആണ് ഉള്ളത് ആക്ടിവിറ്റി എൻഹാൻസ് ചെയ്യാനായിട്ട്?
ആക്ടിവിറ്റി എൻഹാൻസ് ചെയ്യുക എന്നുള്ളത് ബേസിക്കലി നമുക്ക് അത് പല തരത്തിലാണ് അത് നമുക്ക് ചെയ്യാൻ പറ്റുന്നത്. ഒന്ന് ഈ പറയുന്ന പോലെ മ്യൂട്ടേഷൻ, ദാറ്റ്സ് ദേർ. But other than that, mutation means we have to again conduct in various ways. But other than that, we can optimize that phosphate solubilization property also. So the optimization process can be done in various ways by providing different sources, by inducing the organisms in various ways. And then like that, we can have recently we are also working with some of the nano-based method, or nano materials or particles are used for inducing some of the features. So like that, many inducers can be given. That's a very easy method, but the mutation-based methods are also there. That can be but it is not that much practically the optimization will be more easier.
ഓക്കെ സാർ, ഇത്തരത്തിൽ മ്യൂട്ടേഷൻ സ്റ്റഡി വഴി നമ്മൾ പ്രൊഡ്യൂസ് ചെയ്യുന്ന പുതിയ സ്ട്രെയിനുകൾ നമുക്കത് പാറ്റന്റിന് അപേക്ഷിക്കാൻ സാധിക്കുമോ?
ഓർഗാനിസം സച്ച് നമ്മൾ പേറ്റന്റ് ചെയ്യുന്നതിൽ ചില ഇഷ്യൂസ് ഉണ്ട്. സോ ബട്ട് വി ക്യാൻ ഹാവ് അതിന്റെ നമ്മൾ എന്തെങ്കിലും പ്രോപ്പർട്ടി ബേസ് ചെയ്തിട്ടുള്ള രീതിയിലേക്ക് നമുക്ക് ട്രൈ ചെയ്യാവുന്നതാണ്. സോ ദർ ആർ ഈവൻ ദോ സം ലിമിറ്റേഷൻസ് ആർ ദെയർ, we have to present it in a different way. That's the thing.
Okay sir.
വേറെ എന്തെങ്കിലും സംശയം?
സർ, സംശയം സംശയമൊന്നുമില്ലാതെ നമുക്ക് അടുത്ത ഇതിലേക്ക് കടക്കാം.
ഓക്കെ, താങ്ക് യു.
നന്ദി പ്രകാശത്തിനായി ബോട്ടണി ഡിപ്പാർട്ട്മെന്റ് അസിസ്റ്റന്റ് പ്രൊഫസർ പ്രീതികൃഷ്ണനെ ക്ഷണിക്കുന്നു.
Thank you, Gokul sir. A very good afternoon to everyone. Honorable Principal, distinguished resource person, guest, teachers, and dear participants. On behalf of Department of Zoology and Organizing Committee from SVR NSS College, Vazhoor, myself, Dr. Preeta Pillai, Assistant Professor, Department of Botany, feel privileged to propose vote of thanks on this webinar, Microbiology for Beginners, organized by Department of Zoology, Association with IQAC. Before proceeding, my gratitude to everyone, let me show my deep regards to our founder, Bharat Kesari, Sri Padmanabhan, a great reformer who established this Nair Service Society. I extend my respectful gratitude to our beloved principal, Dr. Renuka M.R., for delivering presidential remark and being organized this webinar. In spite of all her busy schedule, she took all the initiative and arranged all the pain. She took all the pain and arranged this webinar. Thank you so much, ma'am, for your interest showing this academic field and motivating us. I have been inspired by the knowledge building webinar presented by Dr. Radhakrishnan E.K, sir, Director, Business Innovative and Incubation Center, and Assistant Professor in Microbiology, School of Bioscience, MG University. Sir, it was very engaging last 1 hour, although after hearing the topic name Microbiology for Beginners, thought that it would be basics only, but you explained very well, sir. You elaborated about whether any particular organism could responsible for disease or not. You also have explained many more things like basic classification to its structure and till research applications available in this area. We also heard some new availabilities of bacteria like rain-making bacteria, electric microbes, many more things we have heard today, sir, new terminology. We also understood about the limitations of unculturable organisms. We understood about the possibilities of sequence-based metagenome, function-based metagenome, human microbiome, plant microbiome, and really surprised to heard that it is quite related to obesity also. It is considered with the obesity. It was surprising, sir. We heard the probiotic, prebiotic, these all are we are learning or listening in the advertisements, but still these all are quite related to with microbes. That is also interesting. And you have elaborated about the plant-based microbes applications. It's in the field of biocontrol, resistance, growth promotions and all. So these all were very interesting, sir. We are on behalf of SVRNSS College, Department of Zoology, and the entire organizing committee. We are really thankful to you, sir. Thank you so much for sparing your time and elaborating this wonderful topic with us. Thank you so much, sir. Next, I take this opportunity to express thanks to Gokul, sir, Faculty of Zoology Department, SVRNSS College, for taking all the pain to organize this webinar. Simultaneously, I would like to take a moment to acknowledge the immense contribution of our dedicated faculty members of this department, Dr. J. Kumar K, Head and Assistant Professor of Department of Botany, Dr. Rajesh M.G, Assistant Professor, and Shruti C.C, Assistant Professor, for their consistent commitment and help for this webinar. Thank you all. I have been impressed by the participants from different parts of Kerala, different colleges of Kerala, who choose to be live with us and attended this webinar with great enthusiasm and made this webinar a great success. Hope this webinar will open your way to choose research in the field of microbiology. Thank you. Thank you all. Last, not the least, our technical support teams, Babin P, Assistant Professor, Department of Mathematics, Remya S. Nair, Assistant Professor, who took initiative and helping us for this webcasting and webinar organization in the term of technical support. Thank you one and all. Thank you for being live with us. Thank you.
Thank you,
Thank
teacher.
you.
ഫീഡ്ബാക്ക് ഫോം ചാറ്റ് ബോക്സിൽ ഉൾപ്പെടുത്തിയിട്ടുണ്ട്, അതെല്ലാവരും ചെയ്യേണ്ടതാണ്. താങ്ക് യു.